of the noise in the background. If not, then thumbs up. If you are listening it, then you can say yes, sir, we are listening this sound. So once again, uh, we will start. Okay, so is the screen visible for everybody? Okay, so let's start again. The liposome-mediated gene transfer, and uh, so good afternoon, everybody. This is second lecture of this day where we are learning about the liposome-mediated gene transfer, and it is a very important chapter for the NEET, MBBS, and other examinations where most of the questions are asked from this, and if the exam will go to the high end. So instead of wasting time on other videos, we will go from basic, and then we will go from the basic to the advanced and the clinical applications. And it's very important how we will end this lecture, and so it's important for you to be with us for a long time so that we can understand it. Okay, now, so oh, in this by the end of this lecture you will understand what are the liposomes, how they help gene transfer, the step by step process, and then we will move towards the advanced and clinical applications, which genes are transferred and when this method is preferred over the other, and how it is used in today's medicine and biotechnology. So first of all, we will understand what are liposomes. What are liposomes? So what are liposomes? Do you know what are liposomes? Liposomes are tiny artificial vesicles. Artificial artificial vesicles. So this is very important to learn about what are liposomes. They are tiny artificial vesicles made up of phospholipid bilayer, made up of phospholipid bilayers. They are similar to the structure of natural cell membrane. They are similar to the structure of natural cell membrane. So this is very important to understand. The structure is same, that is the cell membrane is double membrane, double layered uh, mm, lipid bilayer. In the same way, these liposomes are also double membrane lipid bilayers. So it is very important to understand this. So we usually prepare them by using phospholipids and cholesterol. So what we need? We need phospholipids, and cholesterol and cholesterol, and often add cationic lipids, and sometimes we often add cationic lipids. What are the cationic lipids that we add to them? So the cationic lipids are dotma and dotap. So these are the two cationic lipids that you need to remember if you want to know what are the cationic lipids that we add them. So what is the purpose of adding these cationic lipids? The purpose of the adding these cationic lipids are they carry the positive charge. They carry positive charge. So the purpose is to, because they carry the positive charge on their head. Why we are adding the positive charged cationic lipids? Because it is very simple, because the DNA is negatively charged. Why we are adding it? Because the DNA is negative charge, so we add positive charge so that the same charges attract. They will not repel each other, but they will attract, and the DNA will bind to it. So this is the reason we are adding the positively charged cationic lipids to it. So these positively charged liposomes can bind to the DNA and form the complex. They are called as the lipoplexes. Lipoplexes. So this is important. What is lipoplex? Lipoplex, you can see these are the that I told you. These are the negative positively charged cations. They are added to the, and then this assembly forms the liposome complexes, and then negatively charged DNA or siRNA binding to it to form the lipoplex. So this is the lipoplex, and many these are mostly asked questions in the examination, that what are lipoplexes. In the interviews, people are asking these questions: do you know about the liposomes? What are lipoplexes? And sometimes they ask about the cationic these phospholipids. So the purpose of binding these cationic phospholipids. Okay, so now there are two main ways this can happen. So how can this happen? What is the way to form the liposome? So liposomes simply bind DNA on their surface, and this encapsulations. Sometimes there can be encapsulation of the DNA. That is, DNA is inside the liposome, and that is called the liposome must encapsulate DNA inside DNA inside their aqueous core. Aqueous core. So liposomes sometimes encapsulate DNA inside the aqueous core, protecting their degradation. So this is the first step, and then second main way is liposomes encapsulate the DNA inside their on the surface, not inside but outside on the surface, not encapsulate but simply binds outside the surface. The second step I will explain, that the liposomes simply bind the DNA. They simply bind the DNA. They don't encapsulate but bind is the DNA. Like you have seen in the previous diagram, they bind to the DNA, and this DNA is bound to them and their surface like a coat. So these are the two ways how liposomes are bound to the or lipoplex is formed. So these are the two ways how lipoplexes are formed. So, when these lipoplexes are added to the cultured cell, they interact with the negatively charged membrane and attach on the surface of the then enter the cell either by membrane fusion or endocytosis. So this is the what when these lipoplexes are added to this cultured cells, this cell which need to be cultured, these lipoplexes carry the DNA, and when they attach to it, they bound to the negatively charged membrane, and then attached to the surface, then enters into the membrane there by the process of endocytosis, that is called as early endosome. And inside the cell, DNA is released into the cytoplasm. Like you can see that the DNA is released into the cytoplasm and moves towards the nucleus, and if the plasmid has stable promoter, the gene is transcribed or translated into the protein. So this is a process. Now we will try to understand the step by step process in an easy way, and let us go for a step by step process. So you might be listening the jet airplanes flying. If you not, then how can I increase their sound? Now the sound is more increasing, and you might be listening at how strong the sound is coming. These are the jet planes flying over our premises. So, what are the step-by-step processes? For step-by-step processes, now we will follow the step-by-step process. That is how liposomes are formed or how liposomes are prepared. So, in the laboratory and basic laboratory procedure. So, first is prepare liposome. First is prepare liposomes. Prepare liposomes. So lipids are dissolved in organic solvent, then are dried as a thin film. First is lipids are dissolved in organic solvent, organic solvent, and then dried. They are dried as thin film. As thin film. So first thin film formation takes place. Then film is hydrated. This film is hydrated. It is hydrated, and after that, with a aqueous buffer, aqueous buffer to form liposome. To form liposome. First, it is dried, then it is hydrated to form a liposome. For gene delivery, cationic lipids are mixed in so that liposome carry positive charge. Now, cationic lipids are added to form the positive charge. What are those cationic lipids? I told you earlier that those are dotma and dotap. So here we will write dotma and dotap. These are the two cationic lipids. Now, after that, there is another process that is for from liposome DNA complex to form a liposome DNA complex. Now, liposome DNA complex. Liposome DNA complex. What is liposome DNA complex called? That is called as a lipoplex. So we have to form the lipoplex formation. So purified DNA is added to the liposome. So the purified DNA is added. DNA is added to the liposome, and it will form the lipoplex. Lipoplex. The positive charge on the liposome will attract the negative charge, and DNA will condense and stick to the liposome to form the lipoplex. Then the third is incubate with target cell. Now the third step is incubate with the target cell. And first step is the formation of the liposome. Second step is the formation of lipoplex complex. Then third step is incubate with the target cell. Now the target cell is the cell in which we need to clone or culture these genes. So within the minutes or hours, it will take minutes or hours. The complex attached to the cell membrane, it will attach to the cell membrane, and this cell membrane will take this inside by the endocytosis process, and then the in the endocytosis the DNA will be released and it will go to the nucleus, and then this is called as the fourth step, that is called the entry into cell, where the lipoplex enters the cell by the direct fusion with the membrane or by endocytosis, and they are taken inside then some vesicles called endosomes, and many cationic liposomes formulations and are designed to disrupt the endosomal membrane, helping the DNA to escape into the cytoplasm. The fifth is gene expression. Gene expression. These are the steps. Gene expression is DNA reaches the nucleus. If the plasmid has a functional promoter like CMV, that is a functional promoter, then gene is expressed. So, in simple terms, that liposome-mediated gene transfer uses the cationic liposomes to bind the DNA and form the lipoplex, which enters the cell and delivers the gene for the expression. So, why use liposomes? The advantages and limitations of these liposomes. Why we are using liposomes? What are the advantages and limitations of these liposomes? So this is very important to understand, that you will say why we are reading these liposome mediated transformation gene transfer. If we are having the vectors, if we are having the direct gene transfer, if we are having the this gene gun, what is the purpose? Now let us quickly look and understand the advantages and limitations. So the liposome-mediated gene transfer advantages are relatively simple protocol. This is advantages. The advantages are because they have relatively simple protocol. Relatively simple protocol. Simple protocol. You don't need any big equipment to develop these liposomes like electroporators. Low toxicity. You know the limitations of electroporation is the high toxicity, cell death may occur, and here it is low toxicity. No viral organisms needed, so virus is not needed, so the risk of virus is less. Immune response within the cell is less, so can be it can be used for both in vitro or in vivo applications. In vivo applications. So these are the advantages. What are limitations? What are limitations? So limitations are the transfection efficiency is lower. Yani the efficiency is lower. Transfection efficiency is lower. Efficiency is lower. Stability of the liposome is an issue because they are not stable. Stability is a bigger issue. Is a bigger issue that need to be stabilized. They can form the aggregates, and cationic lipids can sometimes cause inflammation. So the cationic lipids may cause immune response, may cause immune response and immune response. So these are very important points that need to be understood, and this is very important for you. What are the advantages and what are the limitations? Now so you might be knowing that why we use liposomes by understanding their advantages, and sometimes you might be knowing that why preferred over the viral vectors, and why we are not using these in what are the disadvantages of it, and where we need to care take care. So, okay, so this is very easy, and here you have understood this. And now let us go to the advanced. If you are the NEET aspirant or just a basic MBBS student who need to understand this, so for them it is enough, you can leave this video. But if you want to understand it with a depth and want to know its clinical significance, where currently liposome mediated therapy is being done, where currently liposome mediated gene transfer is being done, how scientists are using this gene transfer. So let us go for improving advanced concepts. Now, to make liposomes more efficient, scientists have developed smart and targeted lipoplexes. What are the smart and targeted lipoplexes? What are smart and targeted lipoplexes? Targeted lipoplexes. So scientists have developed these smart and targeted lipoplexes. So ligand targeted lipoplexes. Scientists have developed ligand targeted lipoplexes. Lipoplexes, ligand targeted for lipoplexes. What are ligand targeted lipoplexes? Molecules like antibodies, peptides, sorry, molecules like antibodies, peptides, or growth factors are attached to the liposome surface, and these ligands specifically bind to the receptor and target on the cells such as cancer cell or immune cell. When electric pulse in combination or circulation brings them closer to right cell, then gene delivery becomes cell type specific. So if we want to make the we want to use some antibiotics or antibodies or peptides or growth factors to reach to the cell, so that we can perform the targeted ligand targeted liposomes can be designed, and they are used in combination with the electrophoresis. Pegli- Pegylated lipoplexes. First is ligand targeted lipoplexes. Second is pegylated liposomes. So what are these pegylated liposomes? And first of all, we will understand what is peg. My friend was working on it, and it was polyethylene glycol. So it is added to the surface to form a sheath layer. It forms the sheath layer, sheath teeth layer. It forms the teeth layer, and this helps liposome to avoid detection by immune system and circulates longer in the blood, and which is very useful. So it avoids detection detection by immune system. So is it interesting that you are understanding it at the very advanced level? Immune system, only this channel can give you the advanced level lectures. Now endosome and cap escaping liposomes. So third is endosome escaping liposomes, yeah, or lipids. Some lipid mixtures are specially designed to fuse or disrupt the endosomal membrane and reducing the chance of DNA get trapped or degraded inside the endosome. So you might be getting the question that if the DNA is inside the endosome, that is encapsulated in the endosome or lipoplex, then how will it get released when it is endocytosed? It may or early endosome, then it will become the late endosome. If there is a chance that DNA will remain inside this cell, so scientists have developed endosome escaping lipids, and they get disrupted when they reach inside the cell. DNA is released. So these advanced modifications are exactly what modern biotechnology and gene therapy labs used to push liposome-mediated delivery closer to clinical use. How is it used today in clinical approach? What is the clinical approach these days on the liposome? So the liposome-mediated. So now we are now going to learn about the clinical approach. So clinical approach. What is the clinical approach now? So let us shift to the clinical approach or real world application. Real world application. Applications, real world applications, this is very important, and this is where you will know, if you will follow this video, whole series of this video, then you will understand that this is very important for you, and you need to understand this. So liposome mediated gene transfer is not yet the main first choice method for all gene therapies, but it is important niche where it is strongly preferred. So it is important niche where it is preferred. There it has its own niche where it should be preferred. So where it should be preferred, it should be preferred, strongly preferred in cancer gene therapy. These are the questions. Where liposome mediated gene transfer happens, it is cancer gene therapy. In oncology, liposomes are often used to deliver tumor suppressor genes or immunomodulator genes. So what need to be done? There we have tumor suppressor genes, tumor suppressor genes. तो ये जो ट्यूमर सप्रेसर जीन्स, and or immunomodulatory genes. Immunomodulatory genes. So we have tumor suppressor genes and immunomodulatory genes that need to be transferred inside the cancer cell. That means they will reach the cancer cell and they will suppress its activity. So P53 tumor suppressor gene. What is the tumor suppressor gene? It is P53 tumor suppressor gene can be delivered to P53 deficient cells to restore the normal cell cycle. And cytokines like IL-2, IL-12 or GM-CSF can be delivered to the tumor cell. They are the immunomodulators. Because liposomes can be targeted to the tumor cells via surface ligands and can be injected locally or systematically, and they are attractive for the cancer specific gene therapy. Another is so this is one thing. Another is so now so another is vaccine and immunotherapy. Let us understand vaccine and immunotherapy. Immunotherapy. What is the use of liposomes in it? Liposome based delivery plays a big role in modern DNA vaccine and mRNA type platform. Cationic liposomes, you need to remember cationic liposomes, cationic liposomes, and lipid nanoparticles. Lipid nanoparticles. Cationic liposomes and lipid nanoparticles can carry the DNA and mRNA encoding viral antigens. So, when ingested, these complexes are taken up by the antigen. These complexes are taken up. These complexes are taken up by the antigen presenting cells, which express the antigen and activates the T or B cell. I will explain you. When these are injected, when they are injected inside the cells or in the human blood, these complexes are taken by antigen presenting cells which expresses antigen. Either they express antigen or activate T cell. They activate T cell. They tell to the T cell, oh, get up, there is an enemy, or B cell, and they activate T cell and B cell. So we are delivering vaccine or immunotherapy. This is conceptually similar to the mRNA based vaccine, and this is conceptually similar to the mRNA based vaccines like COVID-19, mRNA vaccine. So, in today's world, liposomes like system are already used in humans, not just in the test tube. So, lung and localized therapy. So, we can do the lung and localized therapies, lung and localized therapies. So, what are the lung and localized therapies? That lipoplexis can be delivered directly into lung, directly into lungs to to treat cystic fibrosis. So these are the very important concepts you need to understand. And another is the for cystic fibrosis, researchers have tried to deliver the corrected copy of CTR gene in the lung using the liposome-mediated delivery. Although viral vectors are often more efficient, liposomal system offers a safer and non-viral alternative, especially when treated dosing is needed. When liposome-mediated transfer preferred over the other methods? Now let us answer the critical question: when do clinicians and researchers choose liposome-mediated transfer over the viral method or electroporation or gene gun? When the safety is priority, so they prefer safety as a priority. When repeat dosing is needed, when repeat dosing is needed. This very important point I am telling you is needed. And when for local target, for local or targeted gene delivery. So this is important for us, and now we have understood what is the role of viral vectors are in conferred when maximum efficiency and long-term expression is needed as a main goal. And physical methods like electroporation gene gun are preferred in vitro research and localized tissue delivery, where you can control the device. So this is very important to understand. One gene gun is needed, and this is gene gun. It is for a in vitro study, and for in vivo study we can use the liposome-mediated transfer. So this is all for today. Please share this video, and we will be this is not for the only for the NEET 2026, but for the MBBS biochemistry MSc medical student. And I have tried to give you a complete understanding of liposome mediated gene transfer by going through the many books and many research papers, and tried to deliver you a best of all wherever you might have found it very difficult, but it's easy